part 3 of this lecture, I'll talk about chemical reactions and enzymes. So chemical reactions are anytime there is a transformation of one set of chemicals to another. They involve the breaking or making of chemical bonds. There are several different types of chemical reactions. For example, here we have chemical reactions that have a synthesis reaction where two individual atoms form a chemical bond to form a new molecule. We have a decomposition reaction where one molecule has its chemical bonds broken to produce two individual atoms. And then we have exchange reactions where one atom is first substituted for another in this molecule or exchanged between different molecules. So the reactants of a chemical reaction are the atoms that go into the chemical reaction, and the products are the molecules that come out. During the process of a chemical reaction, energy can either be used, so there might be overall less energy in the system afterwards, and those are endothermic reactions, or some chemical reactions result in the release of energy, the production of energy, and we call those reactions exothermic. Always in the case, bonds are broken or created during the chemical reaction. So why is it that chemical reactions occur? There are two important theories to understanding this. First is collision state theory. So in collision theory, molecules or atoms they have a certain amount of energy, and so they're randomly shaking around in the systems that they occupy. When atoms or molecules collide with enough force at exactly the right angle, they can break or create chemical bonds between those atoms. So collision theory, you can imagine, predicts that there are several properties of systems that would make reactions more or less likely to occur. For example, if there's a higher concentration of reactants in the system, those reactants are more likely to collide, and that means there will be more productive collisions and more reactions, and we see this in chemistry. Also, if there's more heat or more energy present in the entire system, those collisions will be more energetic. And also, if the molecules are shaped in a way for those reactions or those collisions to happen at exactly the right angle, they'll be much more likely to occur. So collision theory is related to the second theory called transition state theory. So transition state theory is based on this idea that atoms have a certain amount of, or compounds have a certain amount of energy. So here we have the energy of the reactants of the chemical reaction. And here we have the energy at the end of the chemical reaction of the products. If the total amount of energy of the products is more than the total energy of the reactants, then that means that some energy went in to the formation of the products, and that means this is an endothermic reaction. Whereas if the reactants have more energy than the products, then some energy was released, and this is an exothermic reaction. However, in order to get from reactants to products, there has to be some transition compound, some state that happens between the reactants and the products that requires some energy to go over. And so the activation energy is the average amount of energy that a system needs in order to get over this transition state barrier. And the higher the transition state barrier, the more energy that transition state compound has, the less likely the reactants are going to be to go over that barrier and get to the products. Also, you can put more heat into a reaction to make the reactants more likely to go over this high energy barrier. Another thing you can do to speed up chemical reactions to get things over this energy barrier is by using a catalyst. And a catalyst is a compound that speeds up a chemical reaction by lowering the energy of the transition state. By lowering the needed activation energy for a chemical reaction to occur, the reactants are much more likely to go over the hump and become products. So enzymes are biological catalysts, and enzymes are necessary for all of the chemical reactions that occur inside the cell. The reason that enzymes are necessary for life is that all of the chemical reactions, cells are a complex mix of many different chemicals, and they have a need for lots of different chemical reactions to happen very rapidly and very specifically inside the cell to allow organisms to respond to and interact with their environment. So enzymes catalyze chemical reactions in the following way. Enzymes are three-dimensional structures, and they have pockets in that structure called the active site. And the active site has a particular geometry and a particular chemical environment that can bind to a substrate, which is the reactants in an enzymatically catalyzed reaction. The substrate binds into the active site, and the active site provides just the right shape and just the right chemical environment for the chemical reaction of the substrate to occur. Once the substrate is bound in the active site, the chemistry occurs rapidly, converting the substrate to the products, and then the products are released from the active site of the enzyme. In this way, enzymes can catalyze or increase the reaction of many substrates over and over again because the enzyme itself is not changed by the chemical reaction itself. Because enzymes are so important, inhibitors of enzymes are also critical for biological systems. So an inhibitor is a chemical that can prevent an enzyme from catalyzing a chemical reaction. There are two primary ways that chemical inhibitors work. So the first is competitive inhibitors of enzymatic reactions. Competitive inhibitors block catalysis because they bind to the enzyme active site and prevent the substrate from getting into the active site. Non-competitive inhibitors instead bind to the allosteric site, which is just a name for a site on the molecule where the non-competitive inhibitor binds. When the non-competitive inhibitor binds to the allosteric site on the enzyme, it changes the shape of the enzyme so that the active site can no longer catalyze the chemical reaction. So this is the way these two inhibitors can prevent substrate catalysis from happening. So why is enzyme inhibition important? So cells use inhibitors to regulate or to even out, control the activity of enzymes. And so they use inhibitors to prevent too much or too little of a particular product to be made by an enzyme. We also use enzyme inhibitors in medicine to prevent enzymes from pathogens from working. For example, we have an enzyme inhibitor here that is a drug for the virus HIV. So the HIV virus has a particular enzyme called HIV protease. HIV protease is necessary for producing new HIV viruses during the HIV replication cycle. So HIV protease is critical for the virus, and the drug is a competitive inhibitor of the protease. So the drugs that are shown here on the bottom, they have a similar shape as the substrate itself, and so they bind into the active site and prevent the substrate from getting into the active site and undergoing the critical chemistry. And so these HIV drugs are competitive inhibitors of the HIV protease. One important enzymatically catalyzed chemical reaction that we will talk about in depth in the next set of lectures, in the next unit, are condensation and hydrolysis reactions. So condensation reactions are chemical reactions that form bonds by removing a molecule of water from the reactants. So, for example, these two reactants here form a chemical bond between the OH and the H to form a water molecule. And that creates a bond between the two reactants at this remaining oxygen. So that's a condensation reaction. Condensation reactions can also be reversible. In hydrolysis reactions, a molecule of water is added to some reactants to split apart these two individual components. So by adding water back to this chemical bond, it splits these two components into the individual products. That's hydrolysis reaction.